Have you ever just looked around at the sheer scale of physical diversity on our planet and thought, wow, how did this all happen? Like, why is there such a massive variety of physical differences between us humans? And how are these exact same types of adaptations mirrored across the wider animal kingdom? Well, welcome to The Explainer. Today we're digging into a really fascinating source text, a book called Earth, a biography of chaos. And it proposes a radically alternative, biological, and historical model to answer that exact question.
To get a grip on all this, here's our roadmap for The Explainer. We're going to look at the diversity puzzle itself, unpack this idea of a front-loaded genome, check out some rapid animal adaptations, see how that mirrors onto humans, and finally look at how the author says we can actually test these claims.
All right, part 1, the diversity puzzle.
So the central debate here really boils down to two things: timeline and mechanism. When we look at the incredible physical differences across the biological world, the big question is, what biological engine actually produced these changes, and exactly how long did it take? The source text argues that the mainstream scientific consensus might be relying on a biological engine that, well, simply doesn't run fast enough to match the evidence we see.
And this brilliantly sets up the two contrasting frameworks we're looking at today. On one hand, you have the standard model, which is often called uniformitarianism. This view argues that biological diversity was built over deep time, literally millions of years through the super slow gradual process of random genetic mutation and natural selection. But on the flip side, we have the unified theory of geologic history, or UTGH. This model argues something completely different. It says biological diversity happened incredibly fast over just a few generations following a global catastrophic flood event. So instead of slow mutations, the UTGH model points to a totally different mechanism called epigenetic expression.
Which brings us to part 2, the front-loaded genome. This is the proposed engine driving everything.
According to the text, the absolute key to unlocking this whole mystery is this concept of a front-loaded genome. Now, the author suggests that in a pre-flood world, which they describe as having a denser atmosphere and really robust shielding from UV radiation, organisms actually didn't accumulate the genetic damage or mutational load that we see today. Because of that, the founding animal and human populations that survived this global catastrophe possessed genomes with maximum informational fidelity. Essentially, they were completely front-loaded with a massive amount of latent, unexpressed genetic potential. Think of it like a giant library of traits that just hadn't been checked out yet.
So how exactly does that latent potential get unlocked? Well, the text argues it doesn't take millions of years of random mutations at all. Instead, extreme environmental stress acts like a giant switch. Following the catastrophic event, surviving populations were suddenly thrust into radically different super harsh environments, and the severe stress triggered rapid epigenetic expression. Basically, the environment aggressively flipped genetic switches on and off within just a few generations, pulling different physical traits from that deep front-loaded genetic library just to ensure survival. Okay, part 3. Rapid animal adaptations. Let's talk about speed and breadth.
The author uses 3 specific animal families to really show this engine in action. First up, dogs. They demonstrate incredible speed, expanding from a single wolf population to literally every domestic breed we see today in just 300 to 500 years. The text points out that no new genetic information was created, the existing potential was just expressed. Then we have cats, which show the sheer breadth of this expression. They go from a single post-catastrophe ancestor to a massive variety, ranging from a tiny 2-kilogram desert cat to a 300-kilogram Siberian tiger. And finally, horses and donkeys show us the boundary. They rapidly diverged under environmental pressures right up until they hit a hard limit, causing chromosomal divergence where they produce viable but completely sterile hybrids like mules.
But you might be wondering, what was the specific environmental stressor forcing all these rapid changes? Well, the text proposes a very specific thermodynamic aftermath called the steppe window. Try to picture this: a world with incredibly warm polar oceans, but a freezing cold, dark atmosphere just choked with particulates. This massive clash of temperatures drove relentless storms and led to the rapid, explosive expansion of cold, highly productive steppe ecosystems. This sudden, extreme new world was the exact environmental trigger that forced those rapid epigenetic adaptations, creating specialists like the woolly mammoth almost overnight in geological terms.
Moving right along to part 4, the human mirror, let's look at the parallels in adaptation.
Because just like the animals, human populations were also isolated in these extreme new environments. And right here is where the text heavily challenges mainstream human history.
See, the standard model looks at ancient remains, like Homo luzonensis, which was a small-bodied hominin found in the Philippines, and concludes, okay, this must be an entirely separate species of early human, separated from us by hundreds of thousands of years of independent, slow evolutionary development.
However, the UTGH model takes that exact same rule of rapid epigenetic expression we just saw in animals and applies it here. So instead of a totally new species, the UTGH model argues we're actually looking at the exact same human lineage that produced the modern Ida Negro people living in the Philippines today. Because they were isolated on an island with really limited resources, their front-loaded genome rapidly expressed a smaller body plan just to survive. And this is actually a well-documented biological process known as insular or island dwarfism.
So the absolute crucial point here is drawing a direct parallel between human and animal adaptation. The text points out that standard science perfectly accepts that the Channel Islands mammoth was just a normal elephant lineage that rapidly shrank due to island dwarfism. So the author asked a pretty profound question. If we totally accept that biological rule for animals, why do we invent entirely new separate species for humans when they undergo the exact same environmental pressures? Under this alternative framework, it's really just one cohesive biological rule applied across the board.
The author then takes this exact same lens and points it right at Neanderthals. Now, the mainstream model treats Neanderthals as a parallel extinct branch of the human family tree. But the UTGH framework argues that Neanderthals were simply a geographically isolated human population, descendants of those exact same flood survivors who migrated into cold, harsh highland regions. The heavy brow ridges, the robust skeletal frames, the deep barrel chests, the text argues these aren't the markers of a totally different species. Rather, they're the rapid, epigenetically expressed traits of a highly stressed human population adapting to the brutal Ice Age conditions of that post-flood steppe window.
Which brings us to a quote that really captures the heart of this entire alternative model. The diversity was not built, it was expressed. The author is arguing that the vast physical differences we see in human populations today, from skin tones to skeletal structures, they didn't take tens of thousands of years of random mutations to build from scratch. The genetic potential was already sitting right there, preloaded in a single founding population, just waiting for environmental stress to pull it out.
All right, final section. Part 5, testing the claims. Let's talk about falsifiable predictions.
Now, obviously, a scientific model is only as good as its ability to actually be tested. And the text lays out some highly specific falsifiable predictions that the author claims can be verified right now using genetic samples that are already sitting in institutional archives. First off, they predict that shared regulatory sequences will be far more identical across species than deep-time evolution would allow. Second, genetic markers of rapid change, like chromosomal rearrangements, should cluster heavily right after the catastrophe rather than being spread evenly across time. And third, they predict that the epigenetic methylation patterns in modern isolated human populations will perfectly match the specific environmental stressors their ancestors faced right after the flood.
So, the author of our source text throws down a pretty massive challenge to the scientific community. Test the archives. Will future genomic sequencing confirm the standard model of slow mutations over millions of years? Or will the physical evidence ultimately reveal the rapid epigenetic expression of a front-loaded genome triggered by a global catastrophe? It is a fascinating, profound question about our origins, our biology, and exactly how science goes about testing competing models. Thanks so much for joining me on this journey through the evidence. Keep asking questions, and I'll catch you on the next explainer.